Jacob, can you introduce Jay? Thank you.
My pleasure. I'm going to keep this brief because I know that Dr. Zarnetske has a nice long presentation for us. So Dr. Jay Zarnetske is an associate professor of watershed science and hydroecology at Michigan State University, and he's built a career on understanding how water and nutrients are transported and transformed through watersheds. His work spans many scales and ecosystems, from small-scale biogeochemical interactions between groundwater and surface water to large-scale transport of carbon and nitrogen in Arctic watersheds. And we're really excited to have him and his family here coming to Lyon for 10 months starting in September. And with that, I'll let you go, Jay.
Okay, can everybody see my screen okay? All right, yes. And the audio is good? Okay. Well, then let's get started here. So first, thank you so much for the invitation, the opportunity to present some work that I've been fortunate enough to be a part of. And I really look forward to moving to France with my family in the coming year, very soon. I think the visas are arriving at my home today, according to FedEx, so very excited. So today I'm going to try and highlight some of the many spatial gaps that we have when we try to address catchment sciences at many scales. How land is connected to water is a fundamental question that we all must face when studying water and trying to manage our resources. So to get us in the correct mindset and get our brains ready to think about water for the next 40 minutes, I offer you this picture. This is a picture taken by Ansel Adams of the Grand Teton Mountains in the Western United States. So take a moment, look at this photo, and think about what you're seeing. So when we look at this photo, I'm sure, given your different backgrounds, you are seeing different things in the photograph. Your eyes are drawn to different things. You're seeing different aspects. And you might be seeing the light, the beauty of the photo. You might be looking at the mountains and the geomorphology. You might be looking at the plant vegetation patterns. But what I want us all to be thinking about today is the movement of water. So how is the atmosphere connected through precipitation to the mountains? The mountains and the hill slopes are then connected to the rivers, and ultimately how those rivers are connected to our urban centers and our economies. And from that perspective, we have a lot to offer other disciplines. We have an opportunity to do a lot of creative work. And that's really what my research program is built on, is thinking about water as our study organism, in that if we understand the physics and the properties of water, we have the opportunity to work with many other disciplines on major problems. So some examples of our research program working on these socio-technological aspects in New Zealand and France. I'll touch upon a little bit of the work in France today. We work on coastal protection and how plant vegetation can help prevent erosion. A lot of our recent work is in the Arctic on how rapid warming in the Arctic is fundamentally changing that landscape and why that matters. And then a long body of work on watershed carbon and nutrient cycling in the river corridor. So these are interactions between different disciplines, and I try to be the connection between ecologists, biogeochemists, social scientists, and geophysicists. So we'll talk about these three areas today. We'll touch on them in our examples. And the theme of the talk is to be mindful of the gaps, especially the early career people listening to today's seminar. The gaps in knowledge is where you will make the greatest contributions. So find them, go to them, work in them. So I'll provide some examples, both small scale and large scale issues that we have with understanding nutrients and carbon movement through catchments. And the basic one short sentence description of the approach that I present to you today is that physics sets the limits on what can be done, and then the biology gets it done within those limitations. We will use different theory from disciplines today. So you'll see me talk about hydrologic theory, biogeochemical theory, as well as landscape ecology theory. So the two examples where we'll highlight some gaps for you are using streams as sensors of land-water interaction at the catchment scale. And then I will end with a shorter section on groundwater surface water interactions in river corridors, also known as hyporheic zones and riparian zones. So let's start with streams as sensors in the land-water interactions. So as many of you know, streams have always been used as sensors to understand landscape processes. And we've stood here in the stream channel, and we've made many measurements in that, looking at how the water is flowing out of this kind of landscape. And we use that to understand the hydrology, water resources, and ecological questions. We get information about how the concentration of solutes and the amount of change that's occurring in the water chemistry can vary with time. And in this Arctic landscape, how carbon flux is continually increasing over the last 40 years. But the alternative perspective, instead of standing at one place and measuring a time series information, is to spend less time studying the landscape in detail, but to take many easy measurements across space, as shown in these blue dots here. So we can answer different questions and address different gaps when we take this kind of approach. So where and how often do we need to measure water quality to learn how to improve it? All right. With a singular approach as measuring just at one point, we can't answer all of those questions. The other question, and much more detailed question, is how is the structure and the stability of material source and sinks changing at the network scale? So are there particular parts of the landscape that are dominating the chemistry and others that are not? So as I promised this would be an interdisciplinary talk, I wanted to show you that in different disciplines, we have common ways of thinking, but we use different words. So in landscape ecology here, we talk about the extent of a landscape. That would be the study area shown in this square. And then the resolution, or the grain size, is the scale at which we can make particular measurements. All right. So this could be population abundance or the chemistry of the soils. In hydrology, we tend to discuss it differently. We think about catchments or watersheds, and that's our study extent. But we can take catchments and we can break them up into sub-catchments, smaller and smaller resolutions of measurements, and study them as their particular controls on the chemistry of the watershed. So that's what I want to show you now, is the framework that we've been developing to interpret the spatial patterns in stream networks. So here is the introduction to the framework. We have signals generated from watersheds. I have 3 different watersheds here. They are all the same size. They have the same river network and the same measurement locations shown as dots. The one thing that you will see is different is the size of the green squares. Now, each one of those squares represents a strong source or sink location for a particular material, a solute, maybe carbon or nitrogen. All right, and so on the number 1, we have very small patches, and as we move to the right, we have very large patches in terms of the control. These signals get translated from land to water in a way that is predictable. So the grain size that we have here imparts a pattern, and the way that we can interpret that pattern, if we go and measure the chemistry at each one of those dots, is we will see that the variability collapses differently as a function of the watershed or the subcatchment area. So now shown in this plot here is in the small grains, you have different variable chemistry coming together. And once it comes together in the main channel, the signals are interfering with each other and averaging out, making a mixing zone here and a long steady signal as it moves through the rest of the river network. If we have large source and sink controls at a much larger scale, it takes more accumulation area before the signal is well mixed and coming out of the watershed. And in this extreme example of one side versus the other side of the watershed being very different, it takes the whole extent of the watershed before you start to see that convergence, the collapse in the chemistry signal. So we can quantify that. We can also do more advanced work in mass balance approach, where we look at each individual subcatchment's influence on the chemistry. And we can do this in a number of different ways. And we've been doing it in a term what's called subcatchment leverage. And this is basically looking at the role of each subcatchment on the overall mass flux out of the watershed. Okay, and so at the small, basically at the small resolution, each subcatchment is an equal player in the generating chemistry coming out of the watershed. But if you have these big patches, these big controls, then it takes much larger, those larger subcatchments are the ones that dominate the chemistry coming out of the watershed. We can also, with this synoptic spatial sampling, ask questions about the stability of the spatial patterns. So if we go out and we measure again and again and again through the months and the years, we can answer questions of, does that spatial pattern change with time or season? Do perturbations, changes in the landscape matter? Is one watershed always a high source or a low source, or is it constantly changing? So I will walk you briefly through an example here that I think helps demonstrate this. We have two different watersheds represented here. One where the tributaries shown in the different colors of blue here are behaving in an asynchronous way. They're going up and down in terms of their concentration through time. And what comes out of the overall watershed is shown as this black line. That is sort of a mixing of the signal, decreases the amplitude and the variability of what comes out of the watershed. In terms of synchronous watershed, where all the tributaries, all of the subcatchments are changing through time, but they're doing it in a synchronous way. They all go up and down in their concentrations. That signal is preserved and moves out of the watershed with increasing amplitude as it goes out. Okay, so we've got those two different systems. If we can then use that information, we can run statistical analyses on this to talk about how variable it is through time. So this asynchronous one would have a large coefficient of variance. Basically, as you move towards the outlet, you get less and less variability. But in a synchronous watershed, that variability is preserved as you go towards the outlet because each one is moving up and down in unison. The management implication for using this kind of information and determining whether you're in an asynchronous or a synchronous watershed is that it helps you identify where to put your sensors, where to develop your detailed studies. Because if it's a synchronous watershed, then one particular subcatchment is likely representative of what the whole watershed is doing. In an asynchronous watershed, you cannot assume that one watershed is representative of the larger area. We can also ask another question: does the influence of the subcatchment on the larger overall basin and region change through time? And so we can do more analyses on that spatial data and ask, basically, in the simplest way, it's called a rank correlation, but when we go out and measure one watershed again and again and again, does each time, is the subcatchment that have the high concentrations, when we rank them to low concentrations, is it consistent, or is the ranking changing each time we go out there? If it's changing each time, then there's low stability in the landscape. It's highly variable. If it's consistent, then that's very good to know, because it means if its system is stable, is that we don't probably need to measure high frequency. We don't need to invest a lot of effort in those locations because we know that from time to time, things are not changing. So let's put this to use now with a couple examples. The first starting in Brittany. And this is a region that, as most of you know, is dominated by agriculture for nearly 1,000 years in the northwest of France. And we also chose this region because they had repeated synoptic sampling data, which is very rare in the world. And they had across two watersheds, the Quinan and the Rance rivers, they had 56 nested measurements. So shown here in the dots in the two different rivers. And these are quite different landscapes, both dominated by agriculture, and they measured this over 12 years. And in that 12 years, there was changes in the development and agricultural management of the region. And this region, as you may know, has a major water quality issue with nitrate pollution. This work here is spearheaded by Ben Abbott, a fantastic scientist who both Gilles Pinet and myself and others have had the opportunity to work with. He led these analyses. So I give you some quick results here from that study. The thing that surprised us is we had unexpected variance collapse at very small scales in both of these basins. Whether we're measuring carbon, nitrate, phosphorus, the things that we were measuring all collapsed at a reasonably small scale between 18 and 68 square kilometers, all right? The thing that was different was between the two different river networks, the collapse occurred at different scales. So in the Rance, they collapsed at much smaller scales for most of the solutes we measured, shown in the orange bars, and the blue bars represent where we statistically identified the collapse in the Quinon River, okay? And that happened at larger scales. One of the other things that we can identify here is that we found that particular polluted watersheds continued every year to be a strong source of those pollutants. And the watersheds that were not, they were good actors. They did not produce pollutants. They were stable through time. So the management implications here to highlight is that the spatial scales, we can identify mitigation or management efforts to clean up something like nitrate needs to occur upstream at smaller spatial scales than the variance collapse. So at the smaller scales for each of these target nutrients is something that we would need to mitigate at that smaller scale and design our management for that. The other thing is if we have management designs that are associated with total material flux, like loads that are coming out of the basement, we would need to invest our long-term monitoring at larger spatial scales than the variance collapse in order to make sure that we are capturing the integration of both the source and the sink watersheds on that particular solute. We had another unexpected result. We found that there was incredible stability in almost all of the solutes that we measured, all right? And the way we quantify that stability is shown here on the right for a range of solutes from carbon to optical properties, phosphorus, and a range of ions, is basically in the values closer to 1, it means that they're stable throughout many years and across many flows of which the samples were collected. They're closer to 1. This means that despite variability in flow, as I would have predicted and many others, the concentration did not change much in the river network. This stability is really helpful, and it suggests that infrequent monitoring in these two basins is a way that we can proceed. This would reduce the costs and the sampling effort going forward for measuring many of these solutes because of that stability. It's also complementary to more traditional measurements of doing high frequency sensors deployed, put in at the bottom of the large basin. And it also has inspired a lot of new theory and thinking amongst colleagues who are working in this region, including one that just came out at the end of 2020, which is they were trying to explain this stability. And what they did is they came up with a really neat, deep hydrogeologic theory to help explain the stability of a lot of these reactants. So check out that paper. So in summary of this section, I'd like to say that this is a relatively simple framework. You just go collect chemistry across the river network. And it's also compatible then with citizen science or community-based science because you just need to collect water chemistry. There's no advanced field techniques required. And I have to say that the results I showed you here for these two watersheds in France, we expect that to change from place to place around the world in different land uses. So let me show you how we've expanded that work now. So in North America, I'm leading some efforts to do this synoptic sampling in the upper Midwest here, in the Pacific Northwest, which is currently setting records in terms of temperature and has also been largely on fire for the last couple of years. And then in the Arctic, all right, a place that's rapidly warming and changing, a place that's very special to me, and I hope to show you that. But it's also the fastest warming part of the planet. So let's go to the Arctic for our next example. When we think of the Arctic, most people have this impression of what the Arctic landscape looks like. This is a picture I took out of an airplane. I took this picture actually on August 29th, a few years ago. And so we can see even in the peak of summer, you can get snow up there. You can see that there are rivers flowing through the landscape, there's lakes, and that there is effectively almost no visible vegetation. That's because all the vegetation is small and dwarfed due to the cold winters and the presence of ice in the ground. So what you need to know as any scientist, I think, environmental scientists, is that in the Arctic, these landscapes are dominated by permafrost. Permafrost is the word for soil or ground that's frozen for more than 2 years in a row. And in sometimes the ground in these regions of the high Arctic has been frozen for thousands of years. What many of you may not know is that this permafrost landscape in the Arctic represents a massive amount of land mass. 24% of the Northern Hemisphere's total surface area, all right, is underlain by permafrost, as shown here in this map. In that soil, it is estimated that there are massive carbon storage, estimated to be 2 times that of the carbon circulating in the atmosphere currently. We also know now that this Arctic, this northern polar region, is warming at least twice and sometimes 6 times faster than the global mean temperature. So a place defined by ice and cold is warming faster than anywhere else on the planet. And as a result, the Arctic landscape looks like this green, beautiful landscape for much of the summer months. And it's actually increasing. It's getting longer and longer, this what we call the thaw season. So we wanted to use this analysis of spatial sampling and high frequency sensor measurements to identify shifts in the seasonality and understand nutrient fluxes from land to water. So why do we care about that? Well, Arctic rivers, from what we know, they're very hard to study. They're very remote. Very few people have the opportunity or the resources to go there. But those who have been studying the large river basins draining into the Arctic Ocean, and the variability in discharge has not been consistent across the basins. But what has changed is that there's a systematic increase in the solute flux. No matter almost anything that you measure, whether it's inorganics or organics, is increasingly coming out of the Arctic rivers. So what is increasing this mass flux is the big question. There are 2 major hypotheses operating in the Arctic research. One, that it's a diffuse process that's occurring. You have more thaw and more interaction with groundwater, and therefore you just have more coming out of the whole entire landscape into the river network. And we have many lines of evidence to support this, measured at the small soil scales, including geogenic tracers, as I include here some examples. They're going from the soil and into the river network. Another thing that's happening is now that people are taking more effort to study up there, and we're talking with the indigenous peoples of this region, they are noting that the landscape is unraveling. It's falling apart. Land defined by being frozen is now thawing. And so you have all kinds of landscape failures occurring. And this delivers a lot of material into the river network. So is it these point sources that are leading to the increased flux of material out of the large rivers? And this is just another example of some data to prove that that is a significant contribution. But when we study the Arctic sciences and we look across all the disciplines working up there, and we look at how they're studying the system, and we summarize this with one of my postdocs, Dr. Sjogren here, almost all the work is done at plot scales, shown here in this frequency distribution, and at the regional scale, and that at this sub-catchment scale of sort of 1 square kilometer to 1,000 square kilometers, there are far fewer studies. So why do we care about that? That gap, I think, is part of the reason why most studies done at the plot scale are leading to different outcomes in interpretation than those measurements made at the regional scale. We get very conflicting results in terms of when we upscale or downscale our studies. One of the major efforts that needs to be done in the Arctic is incorporating more knowledge about the Arctic into Earth system models. And these models are truly awesome, but they are poorly constrained for those Arctic systems. And they're trying to understand the fate of all that carbon that is in the Arctic. Will it really modify and create a positive feedback in terms of global warming? And now there are system models based on those point measurements primarily is showing that you get between 60 and 240 petagrams of carbon coming out of the Arctic by the year 2100. But as more complicated models that incorporate more ecology into the system, including the vegetation response of the Arctic, you can actually start to see that the Arctic may not yield that much carbon. It may actually just go into the vegetation, and as you grow more larger trees and plants in that landscape. So we've we have, this is what policymakers, they want to know, is it increasing or decreasing? And the models are saying, we don't know. So as a result, the Earth system modeling community has really started to emphasize work in the Arctic, as well as collecting data at this intermediate scale between 1 and 1,000 kilometers squared, so that they can better parameterize these hydrological fluxes and lateral biogeochemical conditions. So we need to collect that data. And this is another argument, a good reason to collect that synoptic data is because it captures that intermediate scale needed for these Earth system models. So I'm going to give you just a couple quick and early results here that show you that we are learning a lot just by taking this new approach. There's 3 different landscapes that we've been studying here. We've got, they're all near each other, relatively close to each other, within about 50 kilometers of each other. There's 2 adjacent watersheds. The one in orange is aquatic watershed. It is a watershed that's tundra, so it has low vegetation all across it, very green, and has a lot of lakes in it. The right next to it is the Hyporheic River, and it is a river network that has almost no lakes in it. And then in the mountains is Trevor Creek, and this is an alpine watershed, so it has very low productivity, very limited amount of vegetation. It's steep, it's colder, and it gets a lot more snow. As a result of these 3 different landscapes, the geomorphic terrain is different, the vegetation is different, the stoichiometry, all right, so the proportions of carbon, nitrogen, and phosphorus in them is also very different. We've been sampling 120 subcatchments across these 3 locations for 3 years now, focusing on, can we detect changes at the beginning of the thaw season when there's very little thaw versus late in the season, like September and October, when there's maximum thaw in that landscape. And the overall goal is to first collect that benchmarking data for Earth system models, and second, using our techniques to identify, are there long-term changes in the nutrient balance across these 3 different watersheds? All right, and so the real hero of analyzing this study for us has been Dr. Ariel Sjogren, shown here, and she's got a series of papers coming out that basically what we're finding is that each watershed behaves differently. And this is important because whether it's got lakes or no lakes or it's alpine, currently Earth system models, almost all of them, do not represent that landscape variability in them. It's just too detailed a question for those models. But landscapes do matter. And I'll show you here. Okay, so we've got the 3 different watersheds, and here's that variance collapse in the chemistry for carbon, nitrate, and phosphorus, just some examples of the data. And we have the Trevor Creek is the alpine watershed in orange, green is the one with the lakes, and blue is the one without any lakes. And what we find is that the steep alpine watershed and the one with lakes have variance collapse at very small watershed scales, and the one with no lakes in a shallow, low gradient landscape takes a much larger spatial scale for the variability to collapse. We also see that it changes depending on the solute that you measure. So we're getting different story than that I said for the French watersheds in Brittany. All right. So the smaller scales matter for lakes and alpine watersheds. We also have learned something incredibly important. There is a representativeness issue that we've detected. The red circled points in each of these is one subcatchment that is included in the blue watershed here, the Hyporheic watershed. And the reason why that's important is that watershed is probably the most well-studied Arctic watershed in all of North America. It has probably had the most amount of money invested in it and the most number of studies published out of that one subcatchment. And therefore, it has a huge influence on the Arctic literature. However, when we put it in the context of how it's behaving in the whole river network, it is an outlier. So how representative is that investigated location of Arctic processes? And we haven't answered the hypothesis yet in terms of, is it a diffuse or are they point scale sources that are controlling this chemistry? But what it does look like with these small patch scale patterns that are emerging is that there's evidence for diffuse sourcing of solutes into the river networks, and that we do see that it is dynamic with time, so different from the French studies. One quick example of dissolved organic carbon and how that stability is changing through the watersheds is Shown here in the Arctic lakes in the early season, basically the colors that you're seeing here show that there's not a lot of variability across the watershed, but late in the season, by the time September comes, the whole watershed has rearranged in terms of who are the high concentrations and who are the low concentrations. And this is important because now it's leading to a new study about the role of lakes in terms of altering the chemistry and the seasonality of lakes. Also important here is that when we measure just at the outlet of this watershed, the DOC concentration and discharge patterns did not show a big change seasonally throughout it. However, spatially, we do see that who's contributing to the overall flow out of the watershed has changed, all right? So this is some good insight to understanding that we would never have understood with just our high frequency single station measurements. So to wrap up this section and move on to the next and last part of the talk here, I just would like to, I found this picture on the sidewalk of H2O Church. So many times we study water, sometimes I maybe worship it, but I would like to preach to you now about some lessons. When we go out and we sample across the watershed, we see things, we learn things that we would not have done if we'd only stood at the bottom of the watershed. So investing that time and effort, we gain a lot of knowledge about the place we are trying to study. So this long-term periodic and synoptic sampling has a lot of value. It's not hard to collect. It just takes a little effort and coordination. And you get to learn things about the representativeness of sites, the spatial scales controlling the major sources and sink processes of solutes of interest, the stability of which a subcatchment may be playing a role on overall water quality conditions. It helps inform where you should monitor, where you can mitigate, impose new management practices, and it really just helps you deeply understand a watershed that you are studying and trying to model. There's brand new results coming out that I just have to tell you about in the Arctic. The Arctic is defined by this cold, dry landscape, but it's actually due to the rapid warming becoming much warmer and wetter. So we've got a real problem in how we're doing Arctic science. We go up there during the summer months, kind of study this small window that you here see when it's very green and warm, and try to make interpretations about how the whole Arctic is working. And this is shown, we've quantified this now for most studies, that most of the work's done between June and August. But we know that there's water flowing and there's things happening in May and in September. And so with the rapid warming of the landscape, this gap is increasing through time. And so we've got to really focus and do the hard work to collect both the early and the late season, what we call the shoulder seasons, where we have not been studying. One quick result on that is to show you that when we start to measure the water flow and the water chemistry in those shoulder seasons, where climate change is predicted to have the greatest effect in the Arctic, we are quantifying now that we were missing about 30% of the annual carbon and nitrate load coming out by only monitoring during this main thaw season area. So that's a huge amount of the budget that we've been missing for decades. Okay, so taking a break to move on to the next thing, which I might have to skip some because I've been going long, is that groundwater surface water interactions in river corridors happen at a huge range of scales, and we've been studying them. And so I'll walk you through some examples and some brand new modeling results of that now. So if nothing else, I want you to learn about river corridors as they are not just the rivers. This is the wrong perspective to think of them as water just moving down a river channel, things flowing in, things flowing out. They're anything but that. Rivers are just the surface expression of groundwater and many complex flow paths. They're very connected to the landscape, and they're connected through the sediment water interface, whether it's flowing through the valley bottom, through paleo channels, or through the subsurface within the channel. So it's a place of extreme heterogeneity given its geomorphic history, and it's also often referred to as the hyporheic zone. The hyporheic word just means hypo and rios, as in Greek origin, below the flow of the river. And this is importantly a hidden ecotone, the boundary between surface aquatic ecosystems and groundwater and terrestrial ecosystems. We often think of this as the river's liver because it plays a big role in altering the biogeochemistry of river corridors. It does this by increasing the residence time of water. It brings it in interact with a lot of solutes and biogeochemically reactive microbial environments. This creates strong chemical gradients, and all this together provides important ecosystem service, including like denitrification, the removal and decreasing of nitrate. So this has monetary value and is of much interest. As a result, over the last 40 years, there's been a big increase in the number of studies working on this groundwater surface water interaction and the hyporheic zone shown in these figures here. The challenge is it spans many scales. And as somebody who's been working on this for almost 20 years, I've worked at now almost all of those scales. So it can go from basically the pore scale all the way up to the subcontinental scale in terms of the research. And we've been working across that range, which makes one's head kind of explode when trying to grasp it. But I just want to walk you through a couple of the major gaps that are known. When we look at the research that's been done on the hyporheic zone, we distribute it by the spatial scale. It's almost all done between 1 meter and 100 meters of river length, and almost nothing at the pore scale or at the large basin scale. So those are the gaps I want to highlight in today's talk, the pore scale and the large scale basins. So I'll start with the small scale because this is some of the latest work and is better developed than the large scale work now. When we look at a river network and we look down in the river channel, we have water flowing, and through the physics of the system, the advection, we transport oxygen as well as solutes down into the subsurface, and then it's discharged back to the river altered. And in this case, the amount of oxygen from the river is high, flowing in, it's consumed and discharged out into the stream water, now depleted with oxygen. But that has been the perspective of, that I've helped publish a lot on, as well as many others. But here I want to show you that there's another scale down at the pore scale where there's a lot of heterogeneity going on. It's not just oxygen rich, it's oxygen rich as well as having some anoxic properties as well. So this has kind of been the paradigm, and I'm a co-author on these papers, so I accept that this is a very useful but also limited interpretation. This is our bulk process domains where the water flows in highly rich with oxygen and number of reactive solutes, nutrients, and carbon. And as it moves through the subsurface before it's discharged, it's transformed. Nitrogen is removed through denitrification, oxygen is removed. And these properties are really useful. And this helps us upscale to the river basin. And that's really the work that we did that helped get us to the whole basin scale work. However, there's a paradox when we use this simple bulk perspective. When I collect measurements, as well as some of my colleagues, we find that in those blue zones, those oxic zones, there are strong signals of anaerobic processes. Here's just one example. As shown in this figure here, the blue region is where it should be oxic. The pore water was measured as oxygen. However, when we look at the metabolites and we look at the products of things that can only happen in the absence of oxygen, we find that that's also abundant in that location, all right? And so how is that? This is the paradox. How do we explain that? And we've been eliciting a theory known as microzones. Why do we care about that? You know, it helps explain why we're finding tons of greenhouse gases coming off of relatively oxic rivers. So this limitation is needs to be addressed. We are now doing this with recognizing that there are lots of anaerobic sites in there and modeling and sampling in this way. And there are many physical mechanisms that can help explain this. So when we look at solutes moving through porous media, we realize that there's a lot of properties that are happening, mechanical dispersion going through the system, spreading out the solutes, but also creating dead zones or places that do not readily exchange with the rest of the water. And those locations are what we are modeling now. We're using pore network modeling, numerical modeling to determine this. And I'm going to move quickly now just to make sure I respect the time. But basically what we find is that the pore network, the physics, the intrinsic properties of the system control where microzones form. And we do get them forming everywhere. And this is a limitation. We've only been doing this with a physical explanation, and the new work we're doing has biofilms included in it. So how do microbes grow and create resources, and how does that change the flow of water? We're testing hypotheses in terms of biofilms promoting these anoxic microzones. And so we've had to develop new models that incorporate the hydraulics, detailed reaction kinetics, and the growth of biofilms within them. And so we now got new feedback on how the flow field moves, including biofilm and bioclogging, now incorporated in these models. And this is work I got to give credit to one of my graduate students, as well as the high performance computing required to do this type of modeling. It's really led to some exciting discoveries. And so when we look at rivers like this, we can now model biofilm growth as well as the chemistry evolution in sandy bedded streams and come up with these detailed and beautiful model results. We can run factorial studies like testing the high and low flow and concentrations with and without microbial bioclogging available in it. And so we can do this for a number of simulations, and I will move through this now to just quickly summarize. But I mean, these model results, first to me, are just beautiful to look at, but also incredibly insightful. And we get things shown here, the distribution of microzones forming and growing. We get those occurring in all of our simulation conditions when we do not have any biomass growing. The story changes, though, once we put biofilms in it. We actually get a lot of change in the flow field, and we get a change in the hydraulic conductivity. It collapses in the system. And so when we don't have biomass growing in the system, we get microzones, as we've been predicting for years. But when we include the microbes in the system in a more accurate, ecologically accurate way, we get almost no microzones forming, and those that do form are not stable, okay? This is because what happens is the biofilm keeps growing and eventually clogs off the system and converts the whole system from having lots of variability to it to just a diffusion dominated system. It's basically a membrane removing all of the oxygen. This is unrealistic, I know this, because sandy beds are mobile and microbes are not consistent, and they die and they live and they die, and they basically are not going to be a permanent barrier. So that's the next adaptation of this model, is to include more realism. So we've got a number of interesting criteria that we've learned here about by including more of the biology and the ecology into the system. And this has been really exciting discovery. We can now, the trick is, how do we measure it? Well, we're using geophysical methods. And so we can use geophysics, which includes lots of batteries, lots of wires, and that's always a challenge to do over water, but it's lots of fun. We can start to measure hyporheic processes at large scales as well, because where limitations of our knowledge, we have got great knowledge now at that small scale, but where we need for management and making predictions at larger scales, we have almost no knowledge, okay? So we have modelers working at that scale, but they have no data to validate their models, or almost none. So students and myself are trying to figure out the first step. How do we measure hyporheic zones at the river network scale? And we've been doing this work. And to quickly summarize, if you've got to invest a lot of effort, the same amount of effort, do not measure lots of things at one point in each reach of the river network. Take smaller, easier measurements and distribute them across the river network to get more information out of understanding hyporheic zones across the river network. We also have a new study that's pretty exciting, which is showing that when you use surface topography to define a watershed, you get a different picture of the water coming to the river network than if you actually look of the groundwater topography. So in our lowland Michigan watersheds, the groundwater shed, shown in light pink here, is very different than the surface topography watershed. And this means we're capturing different sources of pollutants that we would have missed had we just used a DEM or surface model. So in these sections here, I hope I made some clear examples of this gaps are really exciting, and especially for the students and early career researchers, keep calm, identify those gaps, focus on studying those areas. There's a lot of exciting discoveries in those locations. So to summarize with some broad, I think I've left the largest gap for last, and that has to do with the global water cycle. And that recent analysis with Ben Abbott, Gilles, and many others have shown that we're doing a bad job in teaching the global groundwater cycle. And we've updated this now with the latest numbers, but we're also doing a bad job with the teaching of it and the interpretation of it. The places, the gaps where you need to study the most is anything that has to do with groundwater is poorly known. Anything high latitude, so the Arctic, the polar regions. And then in terms of education and the social sciences, we have many misconceptions, because in those diagrams that we see in all of our textbooks, there's almost no evidence of people or anthropogenic influence. And they almost all look like they come out of temperate regions and not representative of the rest of the world. We also recognize that we need a water cycle for the Anthropocene, okay? If we're not representing this diagram accurately and with the good numbers, then we can't possibly manage our limited water resources in an accurate way. So this is the type of work I'm excited to do now that's not at the detail that I gave you before, but much larger scale. There's so much work to do and so many of these crises to address. There's many gaps to fill. And this is what brings me to Lyon next year. I see it as a confluence of opportunities. We'll be working on better integrative projects that really think about river corridors more holistically, with the perspective that water connects us all and we all need to appreciate it. So through meeting the new people, the disciplines, the institutions, and the methods, the data and the policies, I look forward to seeing this confluence of new ideas, new science, and outreach. Many collaborators went into today's talk. I need to thank just the key ones here. I put their colors to associate with how they identify with their home discipline. And you can see just through this project as operating a hydrologist, we get to work with a rainbow of colors, a rainbow of disciplines. It's very exciting. And with that, thank you for your attention. I appreciate the extra time and I welcome any questions.
Thank you very much, Jay. That was very inspiring. And we are really looking forward to having you on board in Lyon for hopefully very interesting and rich collaborations between the institutions, which are all gathered. Several people here in the webinar, which represent the different institutions, and I think they are looking forward to having you from September onward. Is there any question? Just show your hand or raise your voice. Is everybody there? Are you shy? Okay, I can start with one to break the ice, and probably Jake will have the second one. Talking about the paper in the small catchments and the size of the small catchment. I mean, how do you? You say that in Brittany we had measured some stability, but does it have to go with the residence time? So in other words, if the system is really stable in a way, can we infer the residence time by looking at the stability of the system, or is it something that we cannot catch with that?
A great question. I haven't thought much about that. I've thought some about the residence time, in that in places like Brittany, the long consistent land use and the excess nitrogen added to that landscape has created a long legacy, a long history of pollution that is now in the groundwater and will take, you know, decades to millennia to flush out. So with that, yes, the residence time, I think, is part of that signal. So if we find instability in a system, I think that is also indicative of either short residence times, a more responsive system that has less legacy effect, or shorter flow paths and shorter residence times. So this is also, yes, I think this could be useful if coupled with other models to see if the synoptic data and the model of residence times come together. Yeah.
Any question? I don't see everybody, but please raise your hand. Yes, Jean.
Hi, Jay. I was just curious about the work about the decrease of covariance. I was curious if you have the opportunity to test it with only discharge data, because how the variance diminish for discharge, because this would enable us to understand if this decrease in variance is due only to hydrologic processes or to biogeochemical processes.
Yes, yes, I think I understand the question. If we were to look at the collapse and the variance of measuring discharge coming out of the different subcatchments, does this occur?
Exactly, yeah.
Yes, it does. And it varies by region. So the specific discharge, the amount of water that each unit on the landscape contributes, depends where you measure it. So it would be different. It would be different near Lyon. It would be different in Brittany. It would be different in Michigan, but it all collapses. It does collapse. Actually, that paper I cited, the Wood et al. 1998 paper, is addressing that, specifically looking at the discharge coming from many small watersheds. This was him trying to take the theory, which is done more in groundwater, known as the representative elementary area or volume, and tried to say we can do this also at the catchment scale, that the variability eventually collapses at a representative area. And, it's a function of the landscape and the climate that you have in your region. Great question, yes. We have tried to do this in the Arctic and elsewhere, so we are doing this. It's just harder to get distributed discharge measurements than scooping some water into a bottle. That's easy, but discharge is much harder. So. Thank you for the question.
Yes, Jake?
Along all of these same lines with the early part of the presentation, it seems like a useful, important next step is how do you determine if a watershed ahead of time is worth sampling? How is there a way to predict ahead of time what the spatial scale of convergence is, where the variability collapses?
Yeah, I of course, there is ways to do this. You could, if you have models, parameterized models for the basin, you could predict this, I think. You could make different field observations, like walking up the main channel of the river as well, and looking at how the concentration changes. You know, is it going up and down longitudinally? What we would say, you know, more of thinking in terms of like a Lagrangian perspective of if you're following the parcels of water out, is it constantly changing or not? So, but I think this is going out and collecting water is very easy, low technology. It does not require a lot of development time like modeling would. And therefore, I say start with those observations before you invest a year or two developing a process-based model for the same basin. I think it's easy and a good starting place. But yes, we can predict it ahead of time. We can make hypotheses with other approaches.
Jay, in the same way of, since nobody's taking the lead, so I have another question. In Europe, and I know in the States as well, there's a lot of regulatory measurements which are taken by the Water Directive framework in Europe, for instance, and that's been done for the last at least 25 years. The problem is that in many cases, the size of the catchments which is monitored is larger than the one you are dealing with. So the question we have at the moment is that for most of the elements we are, or the sites that we are measuring or having measurements, we are far too downstream to deconvolute the information. So what could be the next step?
Show them this. Instead of investing many thousands of euros into one location and continuing to monitor that one spot. And, you know, often once we make a decision to measure a place, it's very hard politically and financially to move that elsewhere, even if we know it's a bad location. So this, I think, again, is have them, and if that basin is a location of interest for water quality or changing their management, do some synoptic sampling for a couple of years, develop that data, which then you can use to inform, yes, we need to monitor further upstream, further in the watershed, and maybe pick our locations. But yes, there's an expression I use often when it comes to water quality monitoring, is we are governed by the lords of the past. So the people who made those decisions decades ago to monitor that way, given those rules and regulations, still control us today. They limit our options today. So we have to build evidence to prove that we need to change where those locations are occurring.
But you know, the fact that they were occurring on large streams, there was also a kind of undermining ideas is that since we are ran by thresholds and by punishment if we don't comply with the thresholds, people knew from quite a long time that if you go too much upstream in the catchments, you might find very high values in some of the catchments. And in other words, then you don't comply with the requirement. So basically the idea is to, you know, hide a bit the problem under the carpet and go measure downstream where we know it has been mixed a little bit. So this is the key issue, is that we also have to change our minds in terms of monitoring, to move from, you know, being a good student and comply to the teacher in Brussels, or understand what's going on despite the fact that we might be above the threshold from time to time. So that's the big issue. That's the next step.
I agree, and I think something I would like to emphasize out of what you said was people were smart, and they knew where to put the monitoring in order to maybe avoid those thresholds exceeding. So there's people who want to preserve water quality. There's other people who have other motivation, maybe not to preserve the water quality. We have to recognize that there's very smart people on both sides of monitoring water quality, and there's that tension. And this is where the social scientists, not me, but the social scientists need to prevail. They need to have those conversations and recognize that one of my favorite experiences as a consultant was the best people I worked with ended up getting all hired for a lot of money to work for who used to be our clients, the polluters. So when I was working on the site to clean up the environment, the best people I know were paid 3 times as much to use that knowledge to do as little as required to clean up. And we need to recognize that when we come up with these ideas, that there's just as many good and smart people who have different motivation and work towards a different goal.
And I've just, because time is running sadly, but one question a bit provocative about the scaling up of hyporheic zone and its importance. Having had to go through the mill with the riparian zone, trying to understand if it's really important at a larger scale, do you expect that the hyporheic zone would be of some importance in terms of fluxes whenever we go to larger scale?
I think yes, it depends on the specific question. If you're looking at where greenhouse gas, maybe nitrous oxide, is being produced, I think it can be very significant. If it's also important if you're looking at trying to manage for fish habitat or certain protected species. Is it important in terms of regulating the total nitrogen budget across a large river network? I don't know. So it plays a role. It plays important roles. Does it change total mass balance results that you get in large basins? I don't think so. But for very sensitive things like greenhouse gases, I do think it's important.
Because I had to do my coming out with riparian zones. I'm not saying it's not doing anything. I just say that it's sadly not does as much as we expected. Exactly. by just piling up what we measured at a local scale. Because of course we miss quite a lot of fluxes when we go larger. And I think it's not only a multiplication of the sites that we have, but it has a change of paradigm, I suppose.
Yes, yes, that's absolutely true.
Well, I think we are running out of time because the translation will stop very shortly. So I would like to thank you again, Jay, for your very inspiring talk. And all together, we're really looking forward to having you in Lyon from September onward for almost a year. So hopefully with a better virus condition and a good weather as well.
Yes.
So I'll leave the